Thank you very much for your patience. May I now pass the floor to Professor David Baltimore to chair the rest of the summit. Professor Baltimore, please.
Sorry, I hadn't realized it was my responsibility to do this. So we're now going to open the first session on genes, genomes, and genetic variation. The moderator is Catherine Song-Ang Che from the University of Hong Kong. Let me welcome you to moderate this session.
Thank you very much. Can we have, right. I'm Catherine Chia. I'm a professor in the School of Biomedical Sciences at the University of Hong Kong, and it's my pleasure to moderate this first session. Just by way of setting the scene for this particular session, I have just a very few slides just to remind everybody about the tremendous progress and exciting discoveries that have been made over a very long period of time, but that now places us at a very crucial point in the development and the real prospect of not only identifying genes that are responsible for causing disorders, but also reaching a stage where we might actually think of using genomics in order to develop therapies for these disorders. And this slide just summarizes a huge history of how DNA was first discovered, how genes were identified, and how whole genomes have been elucidated in the recent years. And together with that enormous progress in discovery, we can see that starting from the time of the 1980s to now, there has been an exponential increase in not only being able to identify disease phenotypes and linking them to genes, but actually understanding more about how these genes might actually cause the phenotype. So from phenotypes to genes and really back again. And I think that perhaps this enormous progress in understanding what might be responsible for causing a phenotype and then understanding how that gene may actually cause a mutation and that gene may cause a phenotype has been facilitated by technological advances and the enormous information that we may obtain by sequencing genomes and using it for genetic diagnosis. And so this slide just summarizes very quickly how the cost of understanding genetic disease has moved from being horrendously expensive in a very rapid time to becoming very affordable, and that now it may be used in the clinic to diagnose genetic disorders. So in this session, genes, genomes, and genetic variation, we are going to hear 3 talks that sets the stage for a discussion afterwards, starting from monogenic disorders with John Christodoulou from Melbourne Children's Medical Research Institute, and then moving on to understanding the challenges of identifying the contribution of genetic variation in complex disorder from Professor Pak Sham from the University of Hong Kong, And finally, to think about how technology may open up in the future prospects for correcting pathogenic mutations from Professor Dana Carroll from Utah, and of course, the corresponding technological difficulties and issues and the ethics regarding that will be discussed later on in this summit. So without further ado, can I please invite Professor John Christodoulou to give the first talk.
Thanks very much for that kind introduction, Kathy, and I'm delighted to be invited here to essentially set the scene for the current state of play with regards to genomic sequencing technologies and what they have to offer in the current healthcare or provision of healthcare. So during my presentation, I'll briefly set the scene. I'll touch on some of the promises that genomic medicine has, and then I'll move on to talk about the concept of delivering genomic technologies at scale at a national level in terms of being able to offer testing. And then finally, I'll touch on some of the technical challenges that I see that we will be needing to address over the next 5 or 10 years. So I very much work in the rare disease space, in particular in pediatric genetic disorders, and the typical scenario prior to the genomic era when trying to make a genetic diagnosis is this would be the usual set of procedures that would be undertaken to try and reach a diagnosis, starting with a careful clinical evaluation, then looking at some baseline and then perhaps more sophisticated biochemical testing, a chromosomal analysis of one sort or another, often imaging, brain and other organs would be imaged, all of this gathering evidence to help ultimately point to whether there is an underlying genetic disorder, and then if there is, to at least narrow the field on which gene or genes might be responsible. And so the traditional approach was a single gene-by-gene approach to sequencing, which, as you might imagine, is a prolonged, expensive and intrusive diagnostic odyssey that patients have been facing. And so all of this was really transformed by the Human Genome Project, which was a greater than 10-year project that led to the sequencing or delivery of the first draft human genome in 2003 at a cost of about $3 billion. And Kathy has shown a similar slide to this one, which really highlights how one of the major advantages or outcomes of the Human Genome Project was a steady reduction in the cost of sequencing, quality sequencing, and coupled to that was the capacity to be able to build the bioinformatics platforms to be able to analyze big data, big genomic data. And so you can see now that in 2017 and beyond, the cost of a human genome in terms of reagent costs and sequencing costs has gone from $3 billion to around about $1,000 US. And so this has really transformed our capacity to be able to use this technology in mainstream diagnostics. And so, not surprisingly, in the last 6 or 7 years, there has been an exponential rise in the discovery of new disease genes. And in fact, in recent times, this has led some people to speculate, based on available data, that in fact we are starting to see a waning in gene discovery. In other words, perhaps we've reached the peak and there will still be some genes to be found, but the frequency with which they're going to be found is going to be reduced. It really depends on the entity you're talking about, and so On the other hand, this graph shows the gene discovery rate using various platforms in the field of mitochondrial disorders, which is an area that I research in. And at least in the mitochondrial disorders, next generation sequencing, or massively parallel sequencing, as it's also called, still has an increasing gene discovery rate in terms of new disease genes. So in terms of delivering genomic technologies to the healthcare system, what are the kind of promises that we can make? Well, I think firstly, it's really obvious that an early diagnosis or diagnosis can be established earlier on in the diagnostic odyssey. And that immediately has implications in terms of being able to restore reproductive confidence to families that might be at high risk of having a recurrence. In a proportion of cases, this might immediately translate to a targeted therapy specific for the underlying genetic disorder and having an understanding of its biological basis. And certainly in some instances will allow appropriate surveillance for risk factors or complications that can potentially develop, and early interventions where appropriate. The big hope is that genomic technologies and other omics, as they come together, will also help in being able to prevent the big diseases, the complex polygenic disorders. I think we've still got a way to go there, but certainly the building blocks are all there to make this happen. So I'd like to show you a few examples of how genomic technologies really can be transformative. This was a project that was undertaken in Melbourne where 80 infants under the age of 2 years who were suspected of having a monogenic disorder had their usual diagnostic trajectory, and parallel to that they had whole exome sequencing, singleton whole exome sequencing as well. And then some very clever analyses were done to potentially predict what the implication would have been had the whole exome sequencing been introduced early in the diagnostic pathway rather than later on. And what was found was the early introduction of whole exome sequencing would be predicted to have a 5 times higher diagnostic rate than the traditional route, at about a third of the cost of the traditional diagnostic process. And in about a third of cases, this actually led to a change of management, not just in terms of restoring reproductive confidence, but a real change in management one way or another. And following up these cases down the track, the other benefit, of course, was there was a dramatic increase in the restoration of reproductive confidence in those families in which a genomic diagnosis had been made. So all of this really highlights the value of whole exome sequencing at an early stage. Now, currently the traditional time frame for delivering whole exome sequencing is around 3 to 4 months. And so a second study that was undertaken was to examine what are the potential benefits if we were to ramp up the capability and deliver a genomic diagnosis within a 2-week period. And so these data here demonstrate the value. The diagnostic yield was about the same, but the clinical utility was significantly better in those cases where a rapid diagnosis was made. There was an increased cost associated with that in terms of the complexity of being able to deliver a more rapid diagnosis, but the overall cost savings were incredibly dramatic, as you can see in the final row, and this really is important information to be able to provide to policymakers and governments in terms of when it comes to decision making about funding this technology. And so a real-life example where this was applied, I have permission to show this photograph of this young boy and his mother. So this boy presented at 5 months of age with hypotonia and seizures, and over a 9-month period he had lots of investigations to try and identify the cause of his disorder, and he was given a diagnosis of Leigh syndrome, which is a terrible fatal neurodegenerative disorder which is usually fatal within the first decade of life. The genomic sequencing identified him to have a defect in the thiamine transporter, and immediately that meant that we could implement a therapy for him, a very expensive one, thiamine, and the introduction of this therapy ceased further progression of his disease. Equally importantly, however, he had a younger sister who was asymptomatic at the time. She was tested and found to have these mutations in the same gene and was started on therapy before she was symptomatic, and she remained symptom-free 2 years later. So this really highlights the value of early diagnosis in certain settings. So what about moving this from a single clinic into a national arena? And so I come from Australia, so I can tell you about where we're going in terms of the Australian experience. And the first thing to say is the Australian healthcare system is a complex beast. It's a mix of federally funded healthcare initiatives and state-funded initiatives. And the federal government is primarily responsible for funding most testing, whereas the states are responsible for managing the hospitals. And unfortunately in Australia, most genetic testing, and certainly all genomic testing, or virtually all genomic testing, is currently not funded at a federal level, and so it falls to the state governments to fund this. That causes potential problems in terms of equity of access, because different states have used slightly different philosophies and approaches to funding genomic testing. So to try and address this, we were successful in obtaining a 5-year health services research project funding through our National Health and Medical Research Council called and the aim of this is to demonstrate how patients can benefit from the systematic implementation of genomic sequencing technologies at the bedside, in outpatients, in a healthcare setting in real time. And so we're doing this by applying the technology to various rare disease and cancer flagships, and the aim is to identify or validate the clinical utility of this technology in these clinical settings, as well as demonstrating cost effectiveness. And inevitably there will be potential barriers to implementing this technology, sociological, technical, practical, etc. And so a large part of our work is examining those barriers and putting together strategies for how we can deliver this technology at a national level. And a secondary outcome of this project is that we are enhancing the research capabilities and gene discovery capabilities across the country. So Australian Genomics really aims to demonstrate the cost effectiveness and diagnostic efficacy in the delivery of healthcare at scale. And one example, just to focus in on one of the flagships, current estimates are that somewhere between around about 5% of neonatal intensive care admissions, the basis for that admission has an underlying genetic disorder. And I've mentioned that it's been possible to deliver genomic diagnosis in a 3 to 4 month period or even a 2-week period, but that still isn't fast enough in an acute neonatal and paediatric intensive care setting where decisions really need to be made quickly if appropriate treatments are to be implemented. And so one of our flagships is our acute care flagship, where the aim is to deliver an ultra-rapid genomic diagnosis in under 5 days in the sickest of children, those in neonatal intensive care and paediatric intensive care units. The aim being to identify the genetic diagnosis which will then guide management more effectively. And so the processes there are, firstly, one needs to see whether this is clinically doable. And so a large body of work has been involved in developing the clinical and laboratory pathways to make this happen. But equally important is this of relevance from a clinical perspective, and is it cost effective? And so this is a project that's currently underway. We began in February of this year, and our preliminary results so far, we've done 70, and we are using a trio whole exome approach. So the affected proband and both parents are being sequenced simultaneously. And we're very pleased with the outcome so far, where we have so far a 50% diagnostic yield, and listed in the table on your left, yes, on your left, the various genes that have been identified to date in the various categories. And what we're really excited about is through a massive team effort, and this is I should say this is being delivered across about 85% of the country's population. We have a turnaround time in terms of delivery of the blood sample to the laboratory to issuing of a report of just over 3 days. So we think we're really excited about this. Again, in about half of cases, this has led to a change in management, and in about half of cases, we were able to very firmly indicate to families what their risk was of having another affected child, so their reproductive confidence could be restored. So, in finishing up, what are some of the things that we still have as challenges in this current era? We've got fabulous results so far, but there are challenges. The first is, as genomic technologies have been applied beyond the sort of classical phenotypes that people are recognising, two things have been recognised. Firstly, that phenotypes in fact have expanded in terms of the range of phenotypes associated with mutations in particular genes, and current estimates are that maybe up to 1 in 5 genes are going to be subject to phenotypic expansion. And then the second thing that was identified, that perhaps is not so much of a surprise, but was initially, is that in about up to 8% of cases, the phenotype is complex. It's not pure, and it turns out that those individuals, that proportion of individuals, have essentially 2 or more genetic disorders where the phenotypes have become blended. These kind of issues are potentially tractable from a bioinformatics and curation perspective, but at least at this point in time, there's still a great need for manual curation of data and clinical experts in terms of helping to tease apart some of these complexities. A second area that is really is only just starting to become investigated is the whole concept of non-coding variants and their role in primary monogenic disorder. To date, most genomic testing in a clinical setting has been a whole exome-based approach. So that means that the dark matter of the genome is essentially being missed or not looked at. And so it's evident, I think, that a combination of whole genome sequencing and RNA-seq and other approaches, proteomic approaches as well, will start to give us a handle on the non-coding variants and how they might cause primary disease. The third area that really not much work has been done at all is that of oligogenic inheritance. And there are certain areas where that is clearly established, but others where that is yet to be established, and that there's work needs to be done there. And then finally, tandem repeats, beyond those that we currently know of, particularly in non-coding regions, and we are really scratching the surface in terms of what potential implications they might have. So in conclusion, I would venture to say that next-generation sequencing is one of the most disruptive new technologies entered into healthcare in the last decade. Health economic analyses are certainly robust in demonstrating the value of these technologies. There are great opportunities that we face, but certainly risks that need to be addressed from an ethical, legal and social perspective. And so responsible delivery of genomic medicine needs to be tailored to healthcare, and we need to have an early engagement of all of the stakeholders, including government, policymakers and the community. And for me, I have to say it's a great time to be working as a clinical geneticist. And on that note, I'll stop and hand back to Kathy.
Thank you very much, John. I forgot to mention that we will take the discussion and the questions at the end of the 3 talks when our speakers will be up on the pads, on the stage. So welcome Professor Pak Shem from the Center of Genomic Sciences of the University of Hong Kong to give the second talk on complex disorders.
This one?
Yes. Yeah. Thank you very much. Okay, good morning, everyone. I'm going to give a report on genetic variation in relation to common human diseases. So, as we heard from John, single gene diseases are clinically very distinct, and patients are really sort of outliers, are very different from the rest of the population. In contrast, the boundaries of common disorders are much less obvious, and even for discrete conditions like a heart attack, the underlying pathology is often continuous, so in the form of narrowing of the coronary arteries in the case of heart attacks. And the underlying continuous pathology is often determined by multiple genetic and environmental factors sort of acting together. And so this is sometimes called a multifactorial model of disease, and the diagram is an analogy that there are multiple genetic factors and multiple environmental factors, and when the total burden of these risk factors reach a particular value, or so-called the threshold, then the disorder develops. And the relative contribution of genetic versus environmental factors can be determined from twin studies. So we all know that monozygotic twins are often remarkably similar in appearance and personalities and so on. And this is much more so than dizygotic twins, which are 50% genetically the same. And this difference can be used to obtain an estimate called a heritability, which is the percentage of differences that are caused by genetic factors. And this graph shows you that for all common human diseases, the heritability are either substantial or at least modest. And so what we mean by genetic factors are genetic variations in the genome, and the most common form of genetic variation is a single nucleotide change, SNV, or when they are common, SNP, polymorphisms. And these variants arose from ancestral mutational events, and when they have a large impact on the individual, then they cause monogenic diseases. But many variants have more modest or minor effect, and they contribute to common disorders. And these variants that contribute to common disorders can be common or rare. And for common variants, we now have pretty much a complete catalog of them across the world population. And these have been obtained by large-scale projects, such as the HapMap and the 1000 Genome. And the outcome of that is that we now can genotype them very efficiently using SNP arrays. So our latest SNP arrays can genotype more than 1 million SNPs at the same time. Whereas when we study rare variants, because these are so numerous that even despite our large studies and databases, such as EXACT and NOMAD, we still have many that are undocumented. And that's why in the study of rare disorders, rare variants, we need high throughput sequencing technology. Of course, that has progressed and become cheaper, but SNP genotyping technology is still an order of magnitude cheaper than high throughput sequencing. And that is why for the study of common disorders, genotyping is still more popular than high throughput sequencing. So these technological developments have enabled association studies to be done on a whole genome scale. So comparing the frequencies of variants in patients and controls to find variants which may be high risk or protective for disorders. And because it involves testing so many SNPs, there is a risk of false positives, and in order to control the number of false positives to a low level, we needed to adopt highly stringent p-values. The standard is 5 times 10 to the minus 8. And what this means is that we need large samples in order to reach those significant results. And an example of a very large genome-wide association is for schizophrenia. So this study was published in 2014 and combined data from, I think, about 50 studies with a total of 35,000 patients and I think almost 50,000 controls. Sort of this huge international collaborative effort was able to detect 108 loci for schizophrenia, which is a complex, severe mental disorder, and sometimes referred to as the graveyard for geneticists in the past, because it was thought to be so complex and so hopeless. But we now have 108 independent loci and more coming as well. And these sort of outcomes also apply to other common disorders. So this is a graph taken from a paper in 2017 summarizing the status of genome-wide association studies at that time. You can see how the number of discoveries of associations have increased over time and reaching over 10,000 significant associations by the end of 2016, and this has grown even further in the last couple of years to almost 80,000 robust associations for over 300 common diseases. And among these associations are genes that we already are targeting in our treatment of common disorders. The two prime examples are firstly for schizophrenia, because our drugs that we use to treat psychosis are all working on the dopamine D2 receptor. They all are antagonists to dopamine D2. And one of the 108 associated loci was DRD2. And another example is the treatment of hyperlipidemia. So statin can lower blood lipids, and one of the genetic associations for blood lipid level was the gene HMGCR, which controls lipid levels. And one of the observations from that is that the effect size of the natural genetic variant was rather small compared to the effect size of statin. And so a small effect size from the genetic study does not necessarily mean that a gene is not an appropriate target, because the natural variant may have a relatively small effect on gene expression, whereas if we were to deliberately design a treatment, we could design something that has a bigger effect than the natural variant. And this observation that genetic findings may predict drug targets has been taken up by drug companies. So a few years ago, we had a collaboration with GSK, and they looked at their drug development pipelines, and we observed that the drug targets in these pipelines at a more advanced stage tend to have more support from genetic evidence compared to targets that are earlier on in the drug pipeline, the preclinical stage. And this suggests that having genetic support increases the success of drug development. And the list of genes produced, when we have tens or hundreds of genes, can give us insights about the biological processes and pathways that are relevant for a disease. And for psychiatric disorders, an analysis of these genome-wide significant variants have implicated the synapse and the immune system and histomethylation as important pathways for mental disorders. And in our own study on Han Chinese, GWAS schizophrenia, we also identified association with markers on the long arm of chromosome X, a location that contains a MACP2 gene, which controls DNA methylation. So again, sort of indicating the relevance or the importance of gene expression in mental disorders. And so we were quite pleased that the study on copy number variants in schizophrenia also identified chromosome X Q28. And so we have a convergence of common variants detected by GWAS and rare variants, these copy number variants, onto the same genomic locations in the same genes. And the CNV analysis on these 41,000 subjects also implicated genes that were implicated in the GWAS, so namely genes that are important in the function of synapse, genes that are relevant for glutamatergic neurotransmission and dystrophin and interacting partners. And in our studies, we also found associations that are specific to a population. Our study on blood lipids on Asian populations, 47,000 East Asians, including subjects from Hong Kong, reviewed 14 variants that were not found in other populations, either because these variants are rare in other populations or they have a smaller effect in these other populations. And interestingly, when lipid levels were studied, the protective variant for blood lipids, variants that associated with lower blood lipid levels, may be the high-risk variant for another disorder. So this gene, the TM6SF2, is a gene that has a variant that lowers blood lipid levels, but the same variant increases the risk of fatty liver disease. And the mechanism appears to be that this gene is responsible for the transport of lipids from the liver into the bloodstream, so that when the activity of this protein is blocked, is lowered by mutation, then the reduction of excretion of lipids into the blood results in lower blood lipid levels, but then the lipids accumulate in the liver, causing fatty liver disease. So I think this illustrates the importance of understanding the full spectrum of an effect of a mutation before it can be targeted for treatment. And just as a single variant can affect multiple traits, some diseases, different disorders, can be influenced by the same genes. And we demonstrated this using twin studies for schizophrenia and bipolar affective disorder. So this was done almost 20 years ago and recently confirmed by GWAS. So there was significant overlap between the association for schizophrenia and bipolar affective disorder. And this has been extended to a range of other disorders. So you can see here that psychiatric disorders tend to be genetically correlated and also metabolic disorders, different metabolic disorders, also tend to be genetically correlated. And so this genetic sharing between disorders is greatest for diseases that affect probably the same cell type or organs or system. And our genetic findings from common disorders have also been helpful to epidemiologists who have tried to uncover the causal relationship between risk factors and diseases. And this is because one of the problems for epidemiologists is confounding, that even when a risk factor and this disease is associated, it could be because of some confounder. And one solution is to use an instrumental variable which can only affect the disease outcome through the risk factor. And genetic variants provide possibly very ideal instrumental variables, and not only one, but multiple, so that when multiple genetic variants, when used in these Mendelian randomization studies, provide a similar, consistent estimate of the effect between the risk factor, the exposure, and the outcome, then this will give much more confident results, as in this case, for non-high-density cholesterol and heart disease. So the results from both common variants and rare variants, and also our own study, are highly consistent, so indicating that non-high-density cholesterol is indeed causally related to heart disease. And for polygenic disease, every person carries a certain number of high-risk genetic variants, so it's just a question of number. We all carry some high-risk variants for every disorder, and we can now accumulate the effects of all these high-risk variants present in a person to give a polygenic score, and a population can be stratified by polygenic scores into deciles in ascending genetic risk. And for schizophrenia, now we have enough data so that the highest and the lowest decile in terms of the polygenic score now differs substantially in risk, about 8 to 20%, as shown in this graph here. And we can also construct polygenic scores for risk factors such as lipids, so not necessarily directly on the outcome, but also for risk factors, shown here. And it can be applied, say, to cancer screening, because someone with a high polygenic score may reach the risk level which are recommended for the commencement of screening at an earlier age compared to someone whose polygenic risk score is lower. So perhaps needing to have regular monitoring maybe somewhat later, and the polygenic score can be combined also with family history. And recently, we also looked at whether genetic variants predict drug response in relation to schizophrenia. So we identify subjects who responded well and responded poorly to antipsychotic drugs, and we find that there are two gene sets which contain an excessive number of rare damaging variants in the poor responders, and these two gene sets were associated with reduced NMDA-mediated synaptic currents or reduced AMPA-mediated synaptic currents. And of course, these mechanisms are important in memory and learning, which are important for schizophrenia. And current antipsychotic drugs target dopamine, so perhaps it is not surprising that patients with genetic mutation in another system may not respond so well. Okay, I just want to finish with what I think are some of the future trends. So I think we're going to see increasingly large population cohorts which have very comprehensive phenotypic assessments, and assessment of lifestyle, environment, and so on, so that we can sort of look at all these genetic and other factors jointly on a large number of outcomes. I think whole genome sequencing is going to become more popularly used, probably replacing SNP genotyping eventually in a few years' time, and linking up with other data domains, such as clinical data, will be very powerful, and there will be increasing integration with genome functional annotations, such as the ENCODE, epigenome roadmap, and so on, and I think probably greater use of genome functional assays to evaluate the consequences of genetic mutations. So thank you very much for your attention.
Thank you very much, Pac. Our last speaker for this session is Professor Dana Carroll from the University of Utah School of Medicine, and we welcome him. He's a pioneer in genome editing.
All right, thank you, Kathy, for the introduction. I feel honored by the invitation to speak here today. And before I start, I just need to mention that I have modest but some financial interest in a couple of companies that are in the genome editing space, Sangamo Therapeutics and Recombinetics. What I want to do today is offer a perspective on some of the topics that we're going to be looking at in the next few days, and I want to make 3 points. First is, when it comes to the technical aspects of human genome editing, we basically know what we're doing. Regarding the technology itself, we don't yet have adequate control over the outcomes. And when it comes to delivering on the promise of the powerful CRISPR platform, we still have some distance to go. The first International Summit on Human Gene Editing, which has been mentioned before and was held in Washington in 2015, has been compared to the Asilomar Conference, Dr. Baltimore did that, which was held in 1975. And like Dr. Baltimore, I attended both conferences, and I believe the analogy is apt in some ways, but there are also substantial differences. This is true not only of the character of the venue, which was rather rustic in the case of Asilomar and very grand in the case of the first Summit on Human Gene Editing. But there are other differences as well, one of which was that in 1975, we had only a vague idea of the potential applications and possible hazards that might accompany the ability to propagate arbitrary pieces of DNA from essentially any organism in bacteria. Most of the attendees at Asilomar were molecular biologists and geneticists, but there were also a few experienced microbiologists who told us we were crazy to think that we could accidentally or intentionally turn a laboratory strain of E. coli into a dangerous pathogen. Nonetheless, people began to imagine exaggerated disaster scenarios. As an example, in 1976, the city of Cambridge, Massachusetts, the home of Harvard University and MIT, placed a brief moratorium on recombinant DNA research within its borders. Fortunately, these fears and others were unfounded, and in the time since Asilomar, uncounted research advances and many therapeutic innovations, but no illnesses as far as I'm aware, have emerged from the use of recombinant DNA. I don't want to imply that the caution exercised in the early days of recombinant DNA was unwarranted. The fact that scientists assumed responsibility for potential consequences of their novel research capabilities probably enhanced our standing in the public eye. The point I want to make is that the situation is very different today. In 2015 and certainly in 2018, we could point with much more precision to the promise of genome editing in a wide range of applications. We could describe the routes to fulfilling that promise, and we could enumerate potential adverse consequences. In addition, the discussions of technical and social issues involved a broader range of expertise recently than was true for recombinant DNA, at least in the early stages of the recombinant DNA analysis. As a result of the discussions now, I think the public is more focused on the constructive uses of genome editing technology than on the potential dangers. And this is particularly true in the realm of medical applications of somatic editing. The tools of genome editing, zinc finger nucleases, TALENs, and particularly CRISPR, have undeniably revolutionized the practice of genetic research, but bringing these tools to the clinic is a more deliberate process. This is partly due to the requirements for extensive preclinical and clinical testing of any therapeutic before it can be approved for broad use. Nonetheless, the first clinical applications have been launched. According to a US website, all of the editing platforms are being used in current or upcoming trials to augment the effectiveness of cancer immunotherapy with CAR T cells. Both ZFNs and CRISPR are being tested as an ex vivo treatment against AIDS based on the knockout of a co-receptor for viral infection of T cells. All three platforms are being tested for ex vivo treatment of hematologic conditions, sickle cell disease, and beta thalassemia. And there are in vivo applications for attempts to inactivate human papillomavirus using all three platforms. And there are in vivo trials underway using ZFNs to enhance the delivery of therapeutic genes for hemophilia and 2 lysosomal storage diseases. These are among the low-lying fruit for genome editing. They are single gene diseases with well-defined targets, and making the intended changes will essentially certainly have beneficial effects. In many of these cases, the editing does not have to be very efficient, since modest levels will still have therapeutic benefit. The editing is often performed ex vivo, so the challenges of in vivo delivery are avoided. And in most of these examples, the goal of editing is disruption of a genomic sequence. So, full realization of the potential of genome editing is going to require greater control than we currently possess over the cellular processes on which editing depends. And I'm not going to be the last person to show a slide like this in this conference, but the point is that what the genome editing tools do, whether it's CRISPR or the other tools, is to make a targeted break in chromosomal DNA. Everything that happens after that results from DNA repair activities that are inherent to the cell that has suffered this insult to its DNA. Broadly speaking, repair can result in disruption of the targeted DNA sequence, as indicated on the upper right, a gene knockout, or it can result in incorporation of new sequences from a template provided by the experimenter, which could result in the bottom right in a correction of a defective gene. At present, we have the ability to pretty effectively disable individual genes, as in the upper right, but the procedures necessary to make efficient, subtle gene corrections are still elusive, and the efficiency of both outcomes can vary dramatically among cell types. In addition, we're learning that some plausible target cells are intolerant of even a single break in their genomic DNA, which is, after all, the principal thing that we want the genome editing nucleases to do. For somatic applications, we also face the challenge of delivering the editing materials to the desired target tissues and cells. Some tissues are relatively accessible, like the liver, the eye, and the hematopoietic system, but most are not. And conditions involving multiple genes, as Dr. Sham just mentioned, present particularly difficulties. But a genetic disease doesn't have to be multigenic to be problematic for genome editing. Ever since the gene that is mutated in cystic fibrosis was identified by Lapchi Choi and his colleagues in Toronto at the time, nearly 30 years ago, attempts have been made to devise an effective gene replacement therapy for CF. The typical target of these therapies is the lung, since patients die from respiratory insufficiency. But the affected cells are deep in the lung, and no effective delivery system has yet been devised. In addition, by the time respiratory problems emerge, many patients have lost pancreatic function due to the absence of the CFTR protein, and other organs can also be affected. The situation is quite similar with genetic predisposition to breast cancer caused by mutations in the BRCA1 gene. Currently, we have no effective method to deliver molecular therapy to breast cells. Making a genomic correction only in some cells won't be curative, since the remaining uncorrected cells will still have the propensity to turn cancerous. And the same BRCA1 mutations strongly dispose to ovarian cancer and at lower frequencies to other malignancies. These challenges with somatic therapies often turn us to focus from the somatic to germline or reproductive editing, to the elimination of the causative mutation in single-cell embryos and consequently in all cells of the treated individual. It may be surprising that the mechanical operations involved in germline editing are simpler in some ways than what we need in somatic editing. The target cells are readily accessible through in vitro fertilization, and delivery can be achieved by direct injection, as shown here. However, in this case, the concerns over control of both on-target efficiency and off-target effects are much more pressing. This is just an illustration of, just a conceptual illustration of off-target effects. With careful design and testing, all of the genome editing platforms are capable of high efficiency and specificity. In somatic applications, there'll be a limited range of secondary targets that are of particular concern, often sequences associated with cancer, and only the treated individual is at risk. In germline applications, the entire genome is at risk, and it is next to impossible to predict confidently the consequences of particular off-target mutations throughout the processes of fetal, embryonic, and postnatal development, let alone in subsequent generations. Addressing these limitations requires ongoing research, including research with human embryos. Because as I learned yesterday, reproductive editing is happening. We need continuing discussion, as will happen here, of the circumstances in which its application seems warranted, what level of safety must be attained, and what hurdles of regulation must be traversed, and how outcomes should be monitored. Since there's a lot of work to be done, as I've implied, in the laboratory and beyond, I think it's fair to say that widespread benefits, even of somatic genome editing, are still some distance in the future. In passing, I want to mention two areas that are not literally human genome editing, but will likely have a greater impact in the immediate future. One is the use of editing to create animal models of human disease. Because editing makes use of universal processes of DNA repair, it has broadened the range of organisms that can be recruited in such efforts, including larger mammals that are more similar to humans. The other area is agriculture. Some edited food organisms already have hooves on the turf and roots in the ground. Among the engineered traits are improved nutritional value, increased hardiness, and disease resistance, all of which will be of particular value in areas where food security is threatened. So keep an eye on those areas. I want to add one more aspect that I think has to be addressed. How will the benefits of advanced genetic therapies be made available to those who need them around the world? And here I'm going to use sickle cell disease as an example. There are multiple research groups engaged in developing somatic therapies for sickle cell disease and for beta thalassemia. These conditions have several advantages relative to the issues I outlined earlier that are more challenging. These diseases reflect defects in single genes. Exactly the same therapy can be applied to many individuals. Editing can be performed ex vivo in hematopoietic stem cells that are returned to the patient. The clinical manipulations are very familiar. It's essentially a bone marrow transplant. There are prospects for effective editing both by sequence disruption and by sequence correction. And the edits don't have to be 100% efficient, since partial conversion will be therapeutically effective. There are many sickle cell patients in wealthy countries around the world, among African Americans, for example, as noted in the bottom right here. And ameliorating their disease is a worthy goal. The vast majority of sickle cell sufferers, however, are in tropical locations in Africa and Asia. And at present, it's inconceivable that a complex, expensive molecular therapy could be made broadly available in these very high-risk areas. I don't have any bright ideas about how to achieve this, but I have been encouraging smart people, I encourage smart people in the audience, to make distribution of these therapies a subject of future research and development. It would be sad if our powerful technology only increased the disparities in health and in healthcare that exist today. I recently participated in a symposium titled "The End of Illness?" Which reflects what I was saying before, that I think people are optimistic about the applications of genome editing. My assignment was to address the topic, is CRISPR magic? No, CRISPR is constrained by the laws of nature. Still, the prospects are very exciting, even if they're somewhat more distant in the future than we often imagine. Thank you.
Thank you very much, Dana, for a very insightful talk. And we now start the session where we hope the audience will participate and ask questions of our speakers. So I invite John and Pac to come up and have a seat. If the audience would like to come up, there are microphones along the central aisle. If you would like to come up and ask a question directed to the speakers. Who's the brave person who's going to start off? I see somebody coming down. Is there somebody coming down? And when you do, please, would you state your name and your affiliation, please?
Sure.
Thank you.
I'm Matthew Portes from Stanford University. And this goes to the first speaker who is talking about the possibility that the number of genetic diseases that we're diagnosing is going down, or the possibility that's going up. But what I didn't hear you discuss is the possibility that as we find subtle variants of common genetic diseases, we're going to actually find genes that we didn't or diseases that we previously didn't know had a strong genetic basis, but now have a stronger genetic basis. What's your feelings about that? So, yeah, hopefully I made my question clear.
Sorry,
So the question, yeah, okay. So when the disease has severe phenotypes that shows up early in childhood, we have strong suspicions that there might be a genetic basis. But as our tools of diagnosing genetic diseases get better and better, is it possible we'll start identifying more genetic diseases that have subtler phenotypes that show up later in our life course?
Right, yes, absolutely. And perhaps one example that I could give is a childhood disease called Gaucher disease, where mutations in that gene have been associated with an adult onset of increased predisposition to neurodegenerative disorder. So there's no doubt that as these technologies are applied in a broader scale, we will be starting to find not only primary genetic diseases causing a late onset phenotype, but we'll be identifying modifiers of otherwise common diseases that can influence particular genetic changes along the lines of what Pac was talking about, can influence the likelihood of a disorder.
Shi Chong-ing from Hong Kong USD. I have a question for Dr. Carroll. You mentioned about somatic editing, and I think it's a good point you make that you do not need to be 100% knockout to be therapeutically effective. And you also mentioned about sickle cell anemia that has been ongoing work in US. Can you share what's the status of that is, and what kind of dose response curve you will look at? Is it like a linear response, or to the percent of knockout, or biphasic, or sigmoidal, or what?
I don't know a great deal about the clinical aspects of this, but what I understand is that there's sort of a threshold for if you're going to do a correction of the mutation in the beta globin gene. And depending exactly what else is going on, that threshold could be as low as 10 or 20% correction and still provide a good therapeutic benefit.
And may I know what's the best method right now for delivery of the CRISPR in somatic cells?
Well, people are doing this on hematopoietic stem and precursor cells. I don't know that there's a trial yet using genuine bone marrow stem cells, but there are treatments that have been developed using circulating stem and progenitor cells. And there, the cells can be treated in the laboratory, and the delivery of the CRISPR reagents, and people are using the other platforms as well, can be done by delivering protein and RNA in the case of CRISPR, by delivering messenger RNAs in the case of ZFNs and talons, or by delivering DNA containing genes for the nucleases. I don't know, but it seems to me that people have been moving away from delivering genes, since we don't really want persistence of laboratory DNA in the stem cells. But both the messenger RNA delivery and the ribonucleoprotein delivery are more transient, and they seem to be the focus of what people are doing now.
Thank you.
So while we're waiting for the next question, perhaps I can ask as a question of the group of you. It seems to be quite obvious when now we are getting more and more information about genes and their functions in the genome. So if you correct a mutation for a gene, it may have a beneficial effect that seems to be coming out from the talks. It may have a beneficial effect for one particular tissue, but it might actually have additional consequences for another. So how do you think, what should we be doing, thinking about possible therapies in the future as to how to overcome some of these problems? In fact, indeed, sickle cell anemia is an example where it does actually have an advantage for people carrying the sickle cell trait as well. So for people in Africa, if you remove the sickle cell mutation, would you then make them more susceptible to malaria, for example?
Yes, I'm not very knowledgeable about this. I don't think that the sickle mutation is the best defense against malaria. There are others, and there are people working very hard, including the Gates Foundation, on trying to find a more global solution to malaria. So the sickle mutation doesn't mean in a heterozygote or even in a homozygote that you just won't get malaria. So I think that there are better solutions, and the sickle cell disease itself is sort of a high price to pay.
But PAC actually had an example in terms of drugs.
For
the lipid,
blood lipids and fatty liver.
Yes.
Perhaps just to add that as the amount of GWAS data have grew, some people have now started doing what they call phenome-wide association. So instead of looking at one phenotype and searching for genetic association for that genotype, it starts off with one variant at a time and look at all the phenotypes for which there's information, just to see what that one variant does to lots and lots of phenotypes. So I think that may be helpful in sort of screening targets and see which genetic variants have the most favorable profiles and the least adverse effects and more beneficial effects.
And for John, actually, when you are now doing exome sequencing and you identify you've found a few mutations, but actually there are probably many more. And so for that particular patient, maybe that's not the only mutations that's causing the disorder. So how now that you have this kind of information when you're doing exome sequencing on patients that come? How do you manage that?
That's certainly one of the challenges, because there are tens of thousands of variants that will be identified in any one individual, so which is the one that you focus in on? And that, to some extent, revolves around analysis being phenotype-based, and that then, again, is limited by our current state of knowledge about which genes are associated with which particular phenotype. Much of the improvement in our ability to focus in on specific candidate genes or variants to go for has been through very sophisticated refinements in the bioinformatic analysis pipelines that have been developed. But inevitably, or often there will be a situation where you identify a number of variants, you're uncertain about whether they're the pathologically relevant variants, and so the next big bottleneck that we're facing is the so-called functional genomics one. So functional analysis of new disease genes is traditionally a long process, and we need to get smarter in being able to deliver high throughput approaches to functional validation of new genes and new variants in known genes.
Right, thank you. We have a question there.
Thank you. My name is Guido de Wecht, Maastricht University, the Netherlands. I have a question for the first speaker, Professor Christo Dulu. I think we all agree that yes, personalized medicine or precision medicine, whatever term you prefer, has a large benefit and may have large benefit, both for patients and for the population. And likewise, a whole genome or whole exome sequencing, yes, may provide huge benefits. My point is that when you say that the ethical issues linked with whole genome sequencing should be timely addressed, I fully agree. I have a background in philosophy, ethics. I fully agree, but I think a major pitfall would be the equation of whole exome, whole genome sequencing with whole exome, whole genome sequencing and analysis. The sequencing provides raw data, the analysis provides genetic information. And I take your point that yes, there may be quite a few cases, particularly in pediatrics, where whole genome sequencing and analysis provides real benefits, no doubt. My point is that in many cases, it would be sufficient to engage in whole genome sequencing and targeted analysis that may provide real benefits, plus avoid, in many cases, quite serious ethical problems. Can you please comment?
Sure, yeah, and in fact, that's exactly the approach that we take in our centre. So our analysis is very much phenotype driven. So we will perform whole exome sequencing, but only focus in on those genes that are associated with the phenotype for which the initial question was asked. We don't currently subscribe to the American College of Genomics 56 or 59 gene panel that needs to be looked at. We are very focused in terms of the analyses that we do. There are different opinions about that approach, but that's the prevalent approach that we use in Australia at the moment.
Okay, thank you.
Thank you very much. So we have Professor Choi first and then you.
Hi, Lab Choi, Hong Kong. Even though I retired from research, I'm still interested in a lot of things you guys talked about. I think particularly when talk about whole genome sequencing, I must say that even though the technology has improved and the price dropped, but then we're looking a lot of times, you're looking at one gene at a time, comparing the sequence of that gene with the so-called normal version, and therefore you try to draw a conclusion or advice to the parents or patients, especially in the pediatric cases. So I just want to emphasize the importance of a continuing dialogue between the clinician, the genetic counselor, and the family and patients, because as we know that we're now only looking at one gene at a time. There are other genes out there. As you know, there are many mutation carriers don't have a disease. So therefore, it argue really a lot of places is still done as a research protocol instead of standard of care, right? That's point number one. I think John may comment on that. The second one is, again, the same context. Is we're looking at the genome. The gene does not exist in isolation. It exists in the rest of the genome. And then nowadays, we all sort of assume, especially the high throughput sequencing efforts, that our genomes are collinear. Okay, but actually it's not, as you know. And a lot of places there are flips and deletions and whatever. Even though the total number of genes could be similar, but the number of repeats and other things would be very different. And so, of course, a lot of people align their sequence to the reference sequence and then assume that the genomes are collinear. Although people were also talking about de novo sequencing, everything is assembled de novo without a reference. But then when they came to these difficult regions, they had to bypass. Just very simple, the so-called copy number repeats. When they come to that point, they say, oh, we have no clue how to do that, so we rely on the reference or the copy number estimate. So all these actually contribute to the PACS talk. So you give people, you have a multi-genic problem, you give people a score. A score is still based on single genes, and the score for ordinary people, they will not understand what a score means. Although for genetic analysis, it's great so that you have a number to rely on to do the calculation and so on. From the patient side, it came in a score. And it's really very puzzling. So how do you deal with that in the clinical situation? So for the two speakers, I'll ask the third one. Thank you.
Yes. So consumer genomics company have already started to do this, and they usually give some more simplified indication, just green for normal risk and amber for slightly raised and red sort of highly elevated. And so you get a profile, green for heart disease and amber for diabetes and so on. So you know where you need to focus on, I guess, for the consumer genomic company would say, well, you have to pay attention, particularly to diabetes and particularly to whatever they have, and perhaps somewhat less attention. But of course, ideally, you want to reduce your risk to everything. But we all have limited time and so. But yes, reducing it to just a number of bands, I think that's.
And to come back to your first point, absolutely ongoing dialogue is essential. We now recognize that, we know that analysis of a genome or an exome at a particular point in time is just that, a particular point in time. We are discovering new disease genes, and there's been a number of studies that have shown by coming back and re-interrogating the same genomic data a year or 18 months later, you will increase your diagnostic yield by at least another 10%. So we don't know all the genes that are out there that cause disease, and so the beauty of having the sequencing data is you can come back and re-interrogate it at regular intervals.
So,
hi.
Please introduce yourself.
Yeah, my name is Santiago Molina. I'm a PhD candidate in UC Berkeley. I had a question for John. I was wondering what strategies your project is taking to ensure data privacy so if you're advocating for expanded use of genomic data in hospital settings, what strategies have you guys used to kind of ensure that data is being protected?
So you're asking about privacy in genomic data? This is certainly a very important issue, particularly when it comes to insurance and health-rated insurance. I'm sad to say that in Australia we don't have formal legislation, federal legislation protecting the individual, but most recently the insurance industry has self-regulated, in inverted commas, itself, and has put a moratorium on using genetic data for changing risk rating for life insurance, for instance. I'm not so sure that the insurance industry are the people we should be allowing to regulate themselves, but that's a topic for further discussion. But it is important that privacy is maintained in as rigorous a manner as possible. You may have heard that in Australia we're in the process of setting up a national process where an individual's health records are available in a central repository that can be accessed by clinicians who might be looking after them. That has generated considerable controversy, and there are at least 4 or 5% of the population has opted out of having their healthcare data into that system because of these concerns about privacy. So they are incredibly important issues that need to be dealt with, and certainly our country is not alone in this. This is a global issue, and there are activities through the Global Alliance for Genomics and Health, of which Australia is a member, to specifically look at these issues and identify the best ways to deal with maintaining privacy, yet on the other hand, being able to garner maximum information that can help not only in the healthcare of that individual, but of people more broadly.
Thank you.
Charlie Gersbach from Duke University. So I think a lot of the enthusiasm, excitement about genome editing is around the idea of correcting the underlying mutation that causes a disease. And I think the first two talks gave a great highlight of how more and more sequencing is giving us more and more information about different possible mutations that could be targets for that type of therapy. However, if you look at the strategies that are actually kind of leading the path and that are being applied, actually what's going into the clinic first are mostly strategies that don't actually correct underlying genetic mutations, but somehow use gene editing to compensate for it. So some of the sickle cell trials, I think, fall into that category where they're not actually using gene editing to correct the underlying mutations, but rather are going and knocking out a modifier of another globin gene. The same thing is true for the trials in the liver, where they're inserting, for hemophilia and MPS 1 and 2, where they're inserting genes, not correcting underlying mutations. And of course, all of this is because they need to capture large groups of patients all under one single editing strategy. So my question is how you highlighted all of these different possible mutations and more mutations that might be discovered that could be targets. But how do you guys think about how that can be handled in terms of clinical development with strategies that capture more than just a single patient?
I'll have a crack at that. I think the fundamental question there is having a better understanding of the underlying biology, a deep understanding of the biology. And so focusing singularly just on the gene itself gives you a certain level, but as I think Lapchi was indicating, genes don't work in isolation. They're part of, if you like, a system. And so in order to be able to maximize the possibility of being able to use genome editing or other technologies, not just focusing on fixing a mistake, but rather tweaking the system, we've still got a long way to go. And as I said, it ultimately boils down to having a deep understanding of the underlying biology behind the primary gene defect and the genes that interact with that.
I can follow up on that, Charlie, by saying I think one of the terrific things about genome editing, as we've been working with it now for more and more years, is that we're learning so much about biology, and we're learning about both the prospects and the limitations of various systems. And obviously the leading candidates for treatment are taking advantage of situations where the treatment doesn't have to be terribly complex, and it doesn't have to make a specific correction of a mutation that's hard to get at, but is taking sort of an alternative path to something that will be therapeutically beneficial. And obviously two of the things that have to happen for things to get more broadly useful in that respect are, one, our ability to actually correct mutations through some sort of homology-dependent repair, and the other is being able to deliver therapeutic reagents to affected tissues.
So, but the challenge is much greater for complex disorder, right, Pax? So shall we finish off on the complex problem as we finish off the session?
Well, for complex disorders, genetics is only part of it. So, the lifestyle and other factors. But even the genetic part itself is complicated. And as Lachi said, we are now quite mature in studying single nucleotide polymorphisms. But what about the more complex repeats and structural variation? I think we're still a long way. The variance explained by the single nucleotide polymorphism is typically about 50% of the total genetic component. But nevertheless, it's still predictive. But perhaps if we were to target these, targeting a single gene may, either the protein level or the gene level may have a modest effect. If you can target multiple genes, say, 2 or 3 at the same time, and if they are in different pathways, then perhaps there will be synergism. If you, keep on targeting the same, maybe, that target is already fully sort of targeted. But if you, for polygenic trait, if you can perhaps sort of move towards sort of having multiple targets, if that is something possible in the future, that might be an advance.
So on that note, I think there's lots of work for us all as a community to tackle. It's the next Everest, and we shall take a break now, I think. And thank our speakers very much for their insightful